So there's a sign-in sheet back there. People still trickling in here, but I think it's okay. Thank you all for coming. Today we're going to hear from. We're going to have a group talk from our speakers. Need no introduction. We'll hear from our fearless, infallible, and supreme leader, Dr. Gordon Erlebacher, and the vivacious, effervescent, and incandescent Nathan Crock. And so I'll turn it over now to Gordon to get things started.
Okay. So rather than me give a talk, the whole talk, which I normally do, I didn't have time, so of course I imposed on Nathan. But since he did about half the work, it seemed only fair. Okay. So we're going to discuss work that we did last summer. Nathan spent an internship with the people at the Max Planck Institute down in South Florida. This was our initial incursion into the world of biology. And during that period, we had Skype conversations with the people down there every week, both before Nathan went and then during the time he was there for a couple of months. And these are people who worked with experimental procedures, etc. So we were actually working with raw experimental data, which I can tell you is a different world than working with real data. You should be doing this. Okay, so since I'm into game theory, this is like a map of where we're going. So we'll start off with motivation. And then we'll do what people don't normally do, which gives some of our initial attempts of analysis, which pretty much failed. Okay, I think that describing failures is always a good thing. When people tell me how things are done, I much prefer to hear how they got to where they got to as opposed to just the result. I mean, I personally learn much more that way. And then we discuss the current algorithm. So Nathan will be discussing the last algorithm we actually worked with, which actually succeeded, or at least we think it did. And then one slide on applications.
So what we're going to talk about here is the motivation for kind of what brought us into this. So people are always interested in looking at cells and trying to understand what's going on. It's something that's been happening for a long time now. So what we have now is that there's been an advent of new technology and imaging techniques in biology that has given birth to a new need for analysis. So right here I'm just going to show you these quick little videos. So this right here is going to be a video of an astrocyte. Let me see the activity. Right. I was just going to show you so you can't. So the whole point is you can't really see what's going on, right? So this right here is the soma of the cell. It's the cell body. Can you click play one more time? There you go. And so you see, you don't really see too much activity. Well, the reason that is because the old techniques that they had is they would just directly inject fluorophore or dye, whatever it is, into the soma to try and see what's going on. And so that's interesting and all, but all you really see is activity in the cell body itself. And to analyze this, they usually did old school, region of interest techniques where they do ROI averages, region of interest to look at that. And so the analysis was very weak at that time.
Is this a live thing, or is it some tissue that they extracted, or is it a rat on a board somewhere?
I'm not sure. It's from a paper in, I don't know, 2000. There's a recent paper here, and they were comparing, because this new imaging technique that I'm about to show came out in about 2012 or 2013. And so they were comparing the old to the new techniques. And so the new technique, which is a genetically modified technique here, looks as follows. All right, so there's a lot more activity going on here. So the reason this is different is because they actually engineered a technique to modify the DNA of the cell such that the cell produces proteins. And these proteins are then dispersed throughout the entire cytosol of the cell, and they have this curious property that if they come within contact of a calcium ion, the calcium ion binds to the protein, there's a conformational change of the protein, and then if that protein is excited by a photon, it emits a photon itself. And so that's what we actually saw in here, is the calcium is, so the intensity that we see is related to the amount of calcium that is in the area there. So this is the kind of data that we have now. So standard ROI techniques are just entirely insufficient to analyze this. So the next thing we need to talk about is, what are astrocytes? If we're going to be trying to do a lot of analysis on them, we need to understand what they are. So I'm just kidding. We're not really going to look at this. This is miserable, isn't it? But these are actually real astrocytes, okay? I just want you to appreciate sort of the complexity here. This is some of the stuff that's really involved, but this isn't the biology talk. This is something else. So all we need to worry about is this, astrocytes. They basically are involved in the exchange of materials between neurons and capillaries. That's the majority of what we need to be interested in here. The second most important thing for at least motivation is the following. Astrocytes have been shown to play an active role in neuronal dynamics, and everyone's been interested in neuronal dynamics, since Ramon and Cajal in the 1800s. So what we have here is an astrocyte in the center and a couple of neurons around it here. And so the basic understanding or theory of astrocytes is as follows. If there's an activity in a few neurons, this causes an influx of calcium into the astrocyte. The astrocyte then sends information out and modifies all of the neurons that it's connected to. This is just a little graph kind of depicting that. So this suggests that the calcium communication inside of one astrocyte is extremely important in modifying and controlling actual neuronal dynamics in larger populations. So for us to get a larger picture and understanding of the brain and how it works and neurons, we need to understand the role that astrocytes play. And so Gordon's going to talk now about some of the experiments that were done on the data that we obtained.
Right, so when we started, we were confronted with the realities of the experimental world, which is far less clean. Okay, so one typically in biology has to distinguish in vivo from in vitro. So in vitro is when you're analyzing biological data outside the living being, and in vivo means you're actually analyzing the data within the living being. And a lot of work that's been done on astrocytes over the last 20, 25 years, were done in vitro, right? And have found to have nothing, the results have been found to have nothing to do with reality, the reality being in vivo. Okay, so there's a lot of results from the past which pretty much these days are being thrown away. And if you talk to any expert, they will tell you they really have no idea what's going on. In fact, James Schumer, the person we were working with at the Max Planck Institute, told us that if a biologist tells you they understand, they're full of crap. I mean, that's close to a quote. I'm sure I can find my Skype conversation. Okay, so, but it's pretty well known that neurons have what's called tuning effects with respect to orientation patterns. So if you take a graded sheet, okay, where you basically have strips of black and white, okay, at a particular orientation, certain neurons in your receptive field actually respond. And if you actually change the orientation, a different set of neurons respond. I mean, there's overlap between the sets, okay? But you've got this collection of neurons which are all attuned to a particular angle, right? So the idea that what they're working on at the Max Planck Institute is to do the same thing for astrocytes. The question is, are the astrocytes themselves tuned to particular angles? Now, the problem is the astrocytes are receiving information from many neurons, right? And it's not like all these neurons are tuned to a particular angle. Different neurons are tuned to different angles. So the situation for astrocytes is much more complex. Let's see now. Okay, so this is loosely speaking the different orientations that are presented to the ferret, the animal. Okay, so this is experiments with a live animal. And so you have a graded cell. This is just the angle. We present 16 angles at 22.5 degree increments. And then what they're measuring is the calcium, something loosely akin to calcium concentration, not exactly that. Using the GCAM6 experimental technique, which Nathan mentioned. And then from this data, you want to try to infer information about how the calcium data correlates to the angular data. And from that, hopefully derive tuning information. But the initial experiments they did, which is on the next slide, I believe, go like this. This is what they were doing prior to our arrival, okay? And so what they typically do is they take the calcium data, basically 5,000 time steps, 5,000 frames of calcium data, and they do a region of interest, ROI study, which is typically conducted by a combination of principal component analysis and independent component analysis to actually determine what are the regions of maximum activity. So when we started with them, we actually duplicated these results, right? But we found out in the process of doing that the ICAs, the independent component analysis, actually there are many ways of doing this, many more ways than I actually thought. And they have several parameters that you've got to set. And depending on the parameters, these regions would actually change. Now at the lab, they actually use MATLAB routines, where they got preset parameters, and they don't really understand how changing those parameters actually affects these results. So you can imagine I was not a very happy camper. I mean, they write papers and they deduce results, but it's not my particular way of working. However, from these regions of interest, they do the experiments on angles, and then they try to deduce which regions have maximum tuning to which angles. Okay, that you can determine that via some easy analysis, some calculations. And this is region by region. Okay, so this particular region here is actually attuned to this particular angle. Each color is a different angle. And here's pretty much the same analysis done on a pixel by pixel level. So they come up with a technique that allows you to say, for each pixel, for each pixel on this diagram, what is the angle which is it's most receptive to? Now, what do they do with that? I don't actually know. The idea is to develop a model, but the data at this stage is really kind of difficult to analyze and get information from. So one of the tasks we were supposed to do is actually try to get more information out of the calcium video data that they provided us with. Okay, let's see what else comes next. Okay, so if you look on the right, that's basically what we were shown. We were shown this is the animation of what we've got, where the ferret is showed a collection of angles over time, and we were told, okay, from that we would like to get information about the flow of calcium. From that, that's what we're given. That's all we have to work with. Yeah.
Is this one axon or a bunch of them?
This is the result of. This is one astrocyte, so it's like
Astrocyte.
a 300 by 200 grid of pixels, high resolution of calcium in an astrocyte. So the calcium, you could think of it as calcium concentration, but it's not actually that because it's actually what you're measuring is actually influenced by the measurement technique itself. So quantum mechanics people would appreciate this. Okay, so ultimately each angle is shown to the ferret for 16 seconds. It's shown a pattern, and then for 16 seconds the pattern is removed. It's a little bit like between two wines, you taste some water to actually remove the effect so that you can taste the next wine better. Okay, so 16 seconds you show an angle, 16 seconds you remove the pattern, then you go to the next angle, and you just keep doing that. And then the whole experiment is repeated 9 times. So calcium typically is things operate on a slower time scale than neurons. The frequency, the frame rate is at 2 hertz. So there's only one measurement taken every, 2 hertz. That's twice a second. So on a neuronal scale, that's nothing. That's very slow. Neurons operate on a scale of 10 milliseconds. Okay, and then the data is on a grid of around 320 by 250 pixels, and some of us crop the data, some of us don't. So my data later on will be 256 by 256. And the spatial extent is wider than the radius of an astrocyte. You can see, well, you can't see here, but there's one astrocyte here, and there's one astrocyte up here somewhere. Just trust me on that. You can see that if you start averaging the data over 30 frames, then the noise starts canceling out. Okay, next slide. Right, so we were told to get information about the flow, and we were also told try to distinguish the different kinds of flow you may have. Not only the flow, but is the flow diffusive or is the flow convective? Okay, so just to show you, okay, so typically in an astrocyte, in most biological cells, you've got compartments, and these compartments are divided by a lipid layer, fat, in which you've got channels. So these channels, receptor channels, so you've got IP3 receptors or whatnot, and these can also be enzymic, okay, and so these channels can open and close, okay, so these channels open, calcium actually flows in. So when calcium flows in, you can actually get local pockets of high concentration of calcium, and anytime you get a local pocket of high concentration, you get diffusive effects. So a diffusive effect is modeled in 1D by a heat equation, right? So you got a time derivative on the left, and you got a second space derivative on the right. Okay, so on the other hand, you got convective effects. So convective effects in one dimension is where you have a phenomena, and this phenomena at one time, okay, is located somewhere else at a later time. So it just convex along. In 1D, but in 2D or 3D, same thing, like waves in water when you drop a stone. Okay, and those waves, a couple of things. In vitro, which means outside the body, people have noticed in networks of astrocytes, these astrocytes are connected to each other via gap junctions, okay, that the waves can actually propagate through the astrocytes. So that's clearly convective. People have seen that. But I don't think they've seen that in vivo. Okay, like I said, things change in vivo. But what is seen, because you can see it on the videos, once people like Nathan get accustomed to it, okay, along the processes of the astrocyte. So the astrocyte is like a starfish, where the center is a soma, and then you got branches. Those branches are also called processes. And along the processes, you can get calcium moving up and down, right? So those kind of motions are pretty much governed by a wave equation in 1D, right? So du dt plus a du dx is 0, and a is the convective speed, right? So the idea is to make use of some of these concepts to try to get some information out of it. Yes, Tomek?
So could you explain, because there's a difference between the wave and the advection process. So in the ocean you have waves, you don't have advection, right? So do you have advection?
That's correct. So I believe that these experimentalists, I haven't analyzed the math of this stuff because that's not what we're doing, but when they talk about waves, I'm not 100% sure they can answer the question as you posed it. I mean, I notice a difference. You know, a second derivative of time and second derivative of x is a wave. You know, whereas this is one characteristic of a wave going in a single direction. So I cannot answer. I do not know the answer to your question.
So
May
the
I
question whether
Maybe you do.
Whether you have transport of mass or you don't.
Yeah.
You know,
So I can answer that. The leading theory currently is that the calcium ions themselves are not actually traveling all the way down the process. What's actually taking place is something called calcium-induced calcium release, which means you have a small little pocket of calcium and it's being bound to some buffer, and once enough calcium comes to this buffer, he releases those few calcium ions that were connected to him, and this is in a small little radius, and this small little plume then causes another local calcium buffer to plume and plume and plume. So it's a sort of like
it's a wave.
but the calcium ions themselves aren't actually flowing from here to here.
That's what I said, that the wave is different from advection.
Yeah, so the ions are not actually moving.
You don't have advective process.
It's different. Okay, so strictly speaking, one can say there are three types of flow, right? And I can tell you at the end of the day, we can kind of distinguish between diffusion and advection at the end of the day, but we cannot distinguish the two different kinds. I mean, the wave versus the advection, we cannot distinguish that.
Okay.
Right. So what do we want to know? Okay, that's what it says. Right. So the motivation behind all this is get information about the flow of calcium, whatever flow means. Okay. So is the calcium going from the soma to from the inner part of the astrocyte to the outer part, from the outer part to the inner part? Okay. Between the inner part and the outer part, you have diffusive processes, which actually where the flow goes outer and inner at the same time, depending. Okay. There's events happening. If you look at these animations long enough, you can see spikes. Nathan will talk about that. And those spikes can occur in the soma and they can occur in the processes. So what's the relation between the two? Are they occurring independently due to some third event? Or when the soma spikes, does it dramatically make the different processes spike? We also found that when you repeat the same experiment multiple times, the patterns observed are different. Okay, so even though you may understand part of what's happening, the fact of the matter is the phenomena we're not taking care of, we're not taking into account, and so who knows what the effect is. And on top of that, the astrocyte is really three-dimensional, and this data is nothing but a slice through the astrocyte. So there are 3D effects feeding into this from below and above, which we don't see, or at least they're included in the data, but we cannot really quantify short of creating a more sophisticated model. So anyway, they want to get more information than they've got, and the methods they use, the biologists, at least the ones we were working with, are quite primitive, even compared to the primitive methods we're going to show you. Okay. Yeah. Right. So hotspots, lead to diffusive effects. So hotspots are really local, small areas of increased concentration for whatever the reason. Okay. So the minute you get a very small region where you got a high concentration, a high gradient of calcium concentration, right? Automatically that will lead to diffusive effects. Okay. And like I said before, what is the functional relationship between the different events occurring in the astrocyte? That's almost like the holy grail. People kind of know how neurons work. There's activation potentials and signals propagating down the axon, coming in through the dendrites. However, no such theory occurs for the astrocyte. So should I say, there are several theories, with no confirmation. The theories come and go. Yeah. Right. So at the end of the day, people are searching for the astrocyte doctrine. I mean, people understand what's happening in the neuron, but they don't really understand what's happening in the astrocyte. The astrocyte is like astrocyte situations like quantum mechanics before Schrödinger, before the first equation. Okay, and right. So these are our first two attempts. So one attempt is moving Gaussian. So the idea, Nathan was working on that. He came up with the idea of representing the flow by a sum of. If you basically make a plot of the calcium as a function of xy, the idea is to fit that by a collection of Gaussians. So you have to smooth the data, he's going to explain. And then from that, with the proper use of derivatives, the idea was to try to infer some flow. But at the end of the day, that was a dead end. And what I worked on was a method involving cross-correlation. So you have two different signals, and if the two different signals have similar characteristics at different points in time, you can use cross-correlation to extract the data. And from that, I calculated velocity fields. But at the end of the day, that proved to be a dead end when we started comparing that with synthetic data. Yeah.
Right. So my idea was that perhaps, big perhaps, the calcium movement could be described by this concept of moving Gaussians. So to test the algorithm that I'm going to share with you guys here, I made some simple synthetic data, like so. You just have a simple Gaussian moving along a diagonal, and I wanted to see if somehow we could develop some technique to quantify this movement in this flow. So to describe it, I'm going to start off by using 5 frames, but ideally you could use as many frames as you want for the averaging and things that you will see. So it starts off by just taking 5 of the frames of the data. And then what we do first is we transform each of the frames into the frequency space by the discrete cosine transform. We remove the higher modes. This is essentially spatial denoising. Just set them equal to 0 using some coding lingo here. Then, while still in the frequency space, we average together frames 1, 2, and 3 to get a temporal average for frame 2, and 3, 4, and 5 to get a temporal average for frame 4. So this would be considered temporal denoising. We could have done a discrete cosine transform back, an inverse transform, and then did this, but it's immaterial. So once we have the approximation, or the smooth information for the time, the difference between those is going to be an approximation to the temporal derivative at frame 3. So out of 5 frames, we're approximating the temporal derivative at frame 3 using this technique. And then once we have this, we transform this back using the discrete cosine transform back into Cartesian space, physical space here. And we have an approximation to the actual derivative. And once we have this, we downsample it, reduce a lot of the information, and fit it with the 2D cubic interpolant. And we do that so that we can have a smooth C1 continuous surface that we can take the gradient of to try and get some information about the flow. So lastly, once we do all of that, we get this gradient here, which has allegedly has flow information described as follows. So I just got a couple pictures here to show how it worked. So this is the temporal average for frame 2, and this is the temporal average for frame 4. These were F2 hat and F4, or tilde, F2 tilde, and F4 tilde, that I took the difference from to get the temporal derivative. This is what it looks like in 2D and 3D, 2D and 3D, very simple. And then you just take the difference of them, and you get this difference of Gaussians right here. And so if we take the derivative of it, we get these vector fields. And so right here we see a very strong representation of the flow that I was trying to capture. So my idea was, by looking at that, the difference of the Gaussians, that the gradients that we're going to see are going to be the ones associated with the flow. So I just did a simple thresholding to get the longest vectors, and that got the general direction of the flow, and then the average is that red vector there, which is exactly the flow that I was interested in, which works nicely in this very simple case. So the next logical question was, what happens if you have a little bit of noise? So I did the exact same thing again, but adding 10% of noise, and it looks like so. And then I did exactly the same thing again. We have the 2D temporal average and 3D as well, and you can just see the influence of the noise here in these traces along the X, Y, and ZY plane there. But all in all, the amplitude of the signal is large enough such that we're still able to get a strong representation of the flow. So when we take a gradient, we still see here that the majority of information is captured. We do get some small effects due to the noise, but using the same technique and averaging, getting the longest vector again, it's a little bit off the direction we're interested in, but it's still in the general direction. Yeah?
Is the noise you added similar to the signal to noise ratio of the frame data?
It's more. Yeah, it's more. There's more. Yep. So then we have the real data here. I'm sure we can all imagine the tragic dead end that's going to happen once this technique is applied to this guy. So yeah, this is the real data, 5 frames here. So we go ahead and we're going to skip the other frames. Go ahead and just look straight at the flow. So this looks like a disaster. However, we found after trying different techniques that this was slightly less of a disaster than we anticipated, because oftentimes the flow can actually be dispersed and kind of chaotic looking. But I didn't take the time to actually look at the data and quantify whether this technique made sense at that time. I was initially looking into a few different things, which were motivated by this. So the thoughts are, 1, perhaps instead of performing the cubic interpolant on the gradient, I could actually do it in local windows of the actual data itself, so small little pockets. That would be good because I could use, you could have higher order in polynomials if you wanted to get the small little region for more accuracy for the surface that you're fitting. And then allegedly, the difference between these smooth continuous surfaces should provide slightly more accurate results. And while I was looking into this and trying to see, what techniques could be done and what sort of, surface interpolation techniques to use, I found that there already existed an algorithm that does this quite well. It's called the Farnaback's optical flow algorithm. And essentially, what it does is you look at a small region in the image, and you fit a polynomial to that small region, and then you search in a small radius around for all of the polynomial approximations, for which of all the polynomial approximations have the closest fit. So you're basically doing an optimization for trying to say, where did this polynomial approximation here for this region shift to? And then wherever you find the closest fit, you say that that's where this information flowed to. And so this is the technique that I used for optical flow. And so once I did this, I moved on to something else, which I'll talk about a little bit later.
Right, so while he was working on all this, I was working on trying to duplicate the various results that the experimentalists had with whatever their techniques were. And like I said earlier, I didn't like the region of interest techniques they were using. But among the techniques, they were trying to find various types of skewness in their signals, and they were using cross-correlation techniques already, but they were using cross-correlation of signals between two different regions of interest. But right away it dawned on me that if the regions of interest will start changing depending on the parameters of your independent component analysis, it's conceivable that the data, the results they get out of the skewness experiments might also change. Okay, so while that didn't seem to bother them too much, it definitely bothered me. Okay, so let's consider the following. Okay, so we got a reduced grid. This is like a section of the 256 by 256 grid that I use. I crop the data, and let's just consider a grid of 9 points, adjacent to each other, 9 pixels. And now we're going to consider the signals going through those 9 pixels, the calcium signal, in time. Okay, next slide. So here's the calcium signal going through those 9 pixels. This is real data. Okay, so the data's been, I'm pretty sure the data's been blurred. I use 5x5 blur in each frame. I don't do any kind of temporal average. Okay, and so I point the arrow, these arrows point to a particular spike in the signal. I'm calling it spike. Let's just call the spike a small region where the signal goes up and down fast. Okay, and just to show you that in these adjacent pixels, the signals are correlated. I mean, obviously these two signals are correlated, but then there are other parts of the signal which are not correlated. So when you actually multiply those two signals together, you may get something close to 0, but if you multiply this signal times this one slightly shifted so that these peaks align, these peaks will basically form the dominant terms inside the sum, which we call a convolution, and that will actually allow me to actually get velocity information. Okay, next, right? So in 1D, if you have, let's say, a Gaussian, and this Gaussian is propagating from left to right, the Gaussian at this particular point in space and time, okay, is located further down in space and further down in time, right? So if you simply use, a cross correlation, you would multiply the top signal, okay, displaced by this amount in space, and you would multiply displaced by this amount in space and time, and you would multiply it by the other signal, and the signals are exactly the same, and that will give you very high cross correlation. But if you do the same multiplication with a different time shift, you will get almost 0. Now this, of course, is an ideal signal, right? But the beauty of this approach is that because our signal is noisy in time, that the noise will kind of average out, right? Leaving only the terms which are dominant in terms of the cross correlation. So the idea was to actually determine a velocity field based on that. Okay? Oh yeah, hold on. And the formula, this is like the integral formula, which most of you know. And of course, I use a discretized version of this. But then that becomes pneumatic, so we're not discussing that today. Yeah. Okay, so typically, if I follow the biological way, the biologist's way of doing this, or the experimentalist way of doing this, I would take a region of interest that you draw by hand, or you determine by component analysis, independent component analysis, and you would look at the signal in those two regions because they determine that those are the important regions, and then you would calculate the cross correlation or correlations and try to see relationships. But I didn't want to do that. I said, okay, let's take two arbitrary points. Let's start off by calculating the correlation between 2 points which are relatively close to each other, calculate the velocity, and then simply calculate the correlation on every pair of points. In other words, do not presume what's in the data. I don't like to presume. I like to calculate everything and then let the data tell me what's there. Okay? And so what happens is when you calculate this correlation between 2 points in space separated by r, right, we calculate the displacement of the signals, which gives you the maximum cross correlation. And so that displacement in time is tau. And if you divide r by tau, you get a velocity. And because those are particular points, the inference is like the velocity goes from this point to that point. But of course, if I calculate the cross correlation between this point and this point, for instance, I get another velocity. Okay, next slide. Right, so if I calculate the cross correlation between the center point and every other point, I get a velocity vector. This is not the actual size of the velocity vector, right? This is just joins of 2 points. The velocity is r divided by tau. Okay, I could get all the velocity vectors between the central point and all the points around it. And of course, the further the 2 points away, the further the 2 points are away from each other, the less the influence. So in that sense, what I do is I basically draw a circle with a given radius, and I only consider the points within that radius. But I calculate all the cross correlations to get a collection of vectors. And so out of this collection of vectors, now I've got multiple choices. I can either draw all the vectors, or I can actually somehow reduce this collection of vectors into one vector and draw that. You know, and then the other techniques one can use. Okay, and so what I do then is, okay, so I've said this. I'll move on. Right, so what I do is the following. Okay, these are all the successive frames in time. Right, so this is like a video. Time is going from bottom left to upper right. And when I do that, I don't do a cross correlation between minus infinity and infinity in time. I do a cross correlation over n frames, where typically n is either 16 or 32. Right, so I got a discrete sum of 32 terms. And so that effectively, and then I calculate as a function of tau where that cross correlation is maximal. And based on that, I can actually infer the velocity field, you know, between the 2 points. Then what I do, so now I got a velocity field across the entire domain only between, let's say, taking into account, let's say, 16 frames. And then what I do is I advance this window by 1 frame. So I start off from 0 to 16, and then I go from 1 to 17, and then from 2 to 18. So I can actually generate a sequence of velocity fields, out of which then one can actually look at that through animations or static pictures and infer information. Yeah. Right, so these are 2 examples. So you can see these flow fields already look somewhat, well, I'll use the word realistic loosely, but they definitely look more like velocity fields than the pictures that you showed earlier. But of course, I'm doing some form of smoothing in time over 16 frames, whereas he was smoothing in time over 3 frames. So right away, his data would be more noisy, at least the way he did it at the time. So on the left, I'm drawing all the vector fields. You notice that actually, these are the average vector fields, and each point has been reduced to 1 vector. But I also identified points which have special properties, okay? There are some points where the vector field always had the origin at the point and always pointed away from the point. So in other words, if I take these white dots, for instance, you'll notice that the velocity is pointing away in every direction. So at the time, I interpreted that as a diffusive effect, in the sense that the flow originates from somewhere and then diffuses in all directions. Strictly speaking, if you had perfect spherical diffusion or circular diffusion, all these vectors would hypothetically be the same size. Now, why are some vectors missing? Because I introduced into my code, filters which allowed me to determine which vectors. Sometimes the vectors were unreliable or noisy, etc. And I just had various techniques to determine what was noisy, and when it was noisy, I simply did not display it because it just messed everything up. But that also, okay, and on the right, there's a vector field where I don't draw any of these diffusive effects, and one can actually see regions where the field is correlated, right? You can see the flow going this way, then you can see the flow coming down, you can see the flow turning. Okay, so you can actually see patterns, and I've got animations of these things, and they look realistic, but pretty things don't necessarily mean it's correct, right? So we tested this against synthetic data, and and then from that we derived the pros and cons and stopped working on this particular way of doing things. Okay, so starting with, right? So the technique definitely figures out advection patterns. So for instance, I took synthetic data, circular synthetic data, and linear synthetic data, and was able to reproduce the vector fields reasonably reliably. However, diffusion, not so much. If I actually started with a diffusive process, I took a point and and solved the heat equation and created a flow where the flow was expanding circularly, right? The method was not able to reproduce the flow. Right? Now, the difference here, in this particular case, I was able to identify advection patterns which don't necessarily correspond with points of maximum calcium intensity. Optical flows, which we will describe next, always concentrate on taking a pattern and then finding out in which direction the pattern has maximum correlation. And that's typically done with patterns which have maximum intensity. Okay, but in this particular case, you can imagine two signals where the regions of high intensity are decorrelated and the regions of low intensity are correlated. In which case I would identify information regarding flow of low intensity signals, something that the optical flow cannot do. However, we didn't look into that. And then, right, and of course, since we require temporal averaging over 16 or 32 frames, it's clear that we lose information, which means 16 frames or 2 frames a second means 8 seconds, which means that my velocity field is basically, I get the information about the velocity of the flow, but in some sense contained within an 8-second block. And 8 seconds is a long time if you're trying to find out what's going on. Okay, and if I went beyond 32 frames, of course, I got even more averaging, and if I went below 16 frames, I had too much noise. Okay, so there was a limit. Yeah? And at the end of the day, like I said, it didn't do well with diffusion. First and last point is exactly the same. Okay, so next. Right, so what did we learn out of all this? Okay, we learned that at the end of the day, these two techniques were not viable for what we wanted to do, but while we were working on this, we knew we had to work on something different, and Nathan, just so happened, he started looking at the optical algorithms, and he actually found one with the job we wanted. Right, and we also found out that too much temporal averaging destroys information. And of course, you might know this theoretically, but theory and practice don't always match, in my view. Yeah? Right, so at the end of the day, okay, here's a quote from Dimopoulos, and jumping from failure to failure with undiminished enthusiasm is the big secret to success. Okay, at the bottom line, the more you fail, the more you succeed. Okay, so the more frustrated you get, the better you'll be. Okay, next. Okay, so now Nathan will take over.
All right, so given those motivations, we wanted to move on and try and figure out perhaps a more robust technique to answer some of the questions that were posed earlier. So once I realized that I could just look and use somebody else's canned routine to do some optical flow, I was like, why don't I just look and find canned routines to do pretty much everything? My job's not to invent these new algorithms. My job's to give them a tool. And if I can just glue together things that have already been made, why not? So I went in, instead of reinventing denoising, I went and investigated and found that video block matching 3D is pretty much the state of the art in denoising. Anybody who needs to denoise anything, I encourage you to look at that or come talk to me about that because it's great. I'll show you in a little bit. Also, we have to do some alignment with the data because while the ferret is under the microscope, the ferret is breathing, the ferret has a heart rate, right? And so you get this like small little fluctuations in movement in the image. And if we're going to be doing any sort of flow analysis and difference between images, we need these to all be aligned and registered before we do that. So I used the Lucas-Kinect alignment algorithm. And then, as I described earlier, the optical flow using Farnabach's algorithm, which is just polynomial matching in local windows, trying to find out where this local window most likely is highly correlated with in a region around it, based off of Gordon's technique as well. And then, given this, I was able to come up with the two new techniques that allow them to actually analyze the data. And so what I did is I have a way to define calcium flux given this flow, and then a technique very akin to the calcium flux techniques for actually locating and identifying those diffusion events. So, denoising. So this is, clearly you can see over here how successful this algorithm has been. You get the noisy on this side and the denoise on that right side. And the way it works is as follows. This red little square is the region with which they're going to try and denoise. So what they do is they look within the image and within the previous and successive frames for video denoising for statistically similar sub-windows. Because allegedly, this data came from the same recording device, and the noise is allegedly similar. So if they find statistically similar sub-windows, then they stack all of those up in 3D, and they do wavelet decomposition and a lot of other very fancy denoising techniques to try and figure out exactly the type of denoising that needs to be done for this small little sub-window. And this is done over the entire data. And so this was just a canned routine. I went ahead and emailed the authors, asked them for some stuff, and I used their routine quite successfully. So yeah, so that's the before and after. You have the noisy and the denoised, and that's what it looks like there. So how well did the denoising work? This is kind of an important question for those of you who haven't really played with it before. Noise is essentially like an unknown, and as is by the definition, unwanted quantity in your signal. And so if you don't know what it is and you don't know how much it is, how can you get rid of it, right? So the techniques that they do are not objective. There is no go-to technique for doing it. There are always some assumptions or something that needs to be made. The majority of the time, people just presume noise follows a particular pattern, and that is you have high-frequency noise in almost everything. So do like I did with the discrete cosine transform, go into frequency space, remove the higher modes, go back. That's kind of just the de facto technique. But a more robust technique is to actually formulate a noise model based off of your recording devices. So if you understand some of the properties and the physics involved in how you obtain the data, then you can devise a noise model to kind of give you better insight into the type of noise that might exist. So that's actually what I did when I was down there. I had access to their 2-photon microscope and made a little noise model for how much noise was being put into it, from the quantum effects all the way to the photomultiplier tube and everything. So, and this was really important because the denoising technique, the video block matching 3D, it takes in one parameter that's absolutely required, and that's the standard deviation of the noise that you expect to be in the signal. And the noise model gives us exactly that. So I was able to use the noise model to get the standard deviation of the noise that should have been in it and use that for the denoising. And so we did some visual inspection to see its results. So what I wanted to look at is the spatial denoising and the temporal denoising. So what we'll do is we'll take a look at spatial first. So if we zoom into the top there, we have the regular data at frame 74. And it looks like this. This is the denoised using the video block matching 3D. And then this is the go-to de facto technique for denoising for experimentalists. Just hit it with a Gaussian blur and call it a day. Okay, and as we will see, when it comes to denoising, Gaussian blur is just like hitting your data with a sledgehammer. And the video block matching 3D is like going in there with a scalpel and just like removing all the noise in the particular places. It's very nice. So what we do is we take a look here at horizontal slices in the data. So frame 74 here, this top time series is for this horizontal slice through the data, the second one for row 2, 3, and 4, etc. So what I wanted to see here is that spatially, we should see a smothered noise in the periphery where we expect there not to be a strong signal, and the signal should be preserved around the areas where it's important. So here we have red, which is the actual signal. We have blue, which is the video denoised signal, and then green, which is the Gaussian blurring. So as we expect, out here, which is pretty much where a lot of the noise is, because that's pretty much right over here where there is not the soma. This is the soma, and this is the soma. There's not much going on over here. We see that the noise is really high. The Gaussian blur keeps it pretty high, but the video block matching 3D denoising algorithm does a better job at removing the noise than Gaussian blur, while at the same time does a much better job at preserving the signal at the area where we're actually interested in keeping it. And we see this is a common trend in all of the places. I even went across here, right in SOMA, where we expect there to be a lot of intense information, and right here as well, the video block matching algorithm preserved the signal more accurately than the Gaussian blur, and out in the periphery, it did a better job of smothering it around the noise. Okay? So then I wanted to make sure that we didn't mess up anything with the temporal information as well, because we're taking temporal derivatives, we're looking at things in time for the flow, so we looked at the temporal noise as well. Good. And so it's the same thing, except this time we look at the average of the entire time series. So over here we have the regular average of the time series, the denoised average, and the Gaussian blurred average. Go ahead. And what we look at this time are the actual time series for a particular pixel. So we looked at a slice through space before, and now we're looking at a slice through time and looking at these time series. So 1 is the time series at pixel right here in the soma. 2 is 1 directly next to it, just 1 pixel over. 3 is right here and an end foot from the astrocyte. This is actually a blood vessel here where it's extracting nutrients to keep it alive. And 4 is out here in no man's land where there should be all kinds of noise. And so, again, as we expect, the video block matching 3D, the blue curve, does a much better job at preserving the signal in the soma where information is important, as well as the pixel next to it. I did this because Gordon was interested in like the correlation between pixels close to each other and how the denoising was affecting, so you can see that. And same thing I said here at the end foot, and then out here where there's lots of noise, again, the video block matching algorithm in general does much better than the Gaussian blur. Somehow it just introduced artifacts of noise there in time. So all in all, spatially and temporally, the video block matching algorithm did a much better job at removing noise in our data. And so this, for us, was enough evidence to keep it and move on. So now what I can show you is the results. So the noisy data on the left and the denoised data on the right. And so once we had this denoised data, it was the flow results were much more robust. We weren't getting quite as many artifacts and random vectors out in the random periphery. And we can take a look at that soon. So next is so go ahead and click play over there. So this is what the data looks like before alignment. Just as you can see how it's jumping around all jittery like that? That's due to respiration, that's due to the heart rate of the ferret, and unfortunately maybe some experimental error. Experimental's bumping the table or something. So that's just unacceptable. So we needed to use the Lucas-Kinect algorithm to try and align it here. And so the Lucas-Kinect algorithm works as follows. what it'll do is it'll find certain landmarks and features in the data, and what it'll do is it'll use one frame as a reference frame and say, I need to align everything else to this frame. So it finds landmarks in this one data, and then looks through everything else to find those similar landmarks, and then looks for the displacement of those landmarks to the image, and then shifts it and translates it to accomplish that. It's actually another approach to optical flow, but the optical flow algorithm we're working on is more robust. It's just for alignment. So next, we can take, you got to click off the video. And then next. There you go. So now we look at it after alignment. You play that video for me. So this is what it looks like after alignment. It's nice and steady. You sit there, you see a couple of pulses out in the processes. The information is really rich here. This also happens to be averaging as well, because they did this experiment 8 times, aligned it, and then averaged everything together. I apologize, I couldn't get my hands on the purely aligned data, but it gets the point across. There's nowhere near as much movement going on here, so the alignment was necessary. All right, so go ahead. Click off the video. All right, so now we've got the data has been denoised and the data has been aligned using Lucas-Kinect algorithm. So now what we need to do is determine the optical flow. So just in case people aren't familiar with what optical flow is, we'll talk about it. So play this video for me. So this is what optical flow is here. Basically, it infers motion from pixel translations. Okay, and so the problem statement is formulated as follows. Pretty much anyone doing optical flow uses this fundamental theoretical problem statement to solve it. So we have I, which is the pixel intensity at location x, y, and frame t. So at any point along here, you've got some x, y location and some intensity there for the information. If we do a a Taylor expansion for this, we can get this expression here, and this represents the intensity and then some small perturbations in space and time. So what we assume in optical flow is that this extra information is 0. What this means is that the intensity information at time t and the intensity information at time t plus delta t is the same. So that means in frame 1, if I'm standing right here, and then frame 2, I jump over here, the intensity of my shirt is the same as it was here as it is here. So then you can actually look for this intensity being translated to some other location in the next frame. And so this is the problem that we have to try and solve this statement right here. So what this turns into is something that most people are probably familiar with. You divide through by dt in this expression, and you get the known spatial and temporal gradients and the original image itself. And what we want to know are u and v. What are u and v? These are the actual flow information for one particular pixel. It's actual translation. And so solving this problem is the root of all optical flow algorithms and techniques. All right. So I applied the Farnabax's optical flow algorithm to our data. So can you play this one over here for me first? This guy? So in this video, you're going to see a spike in the soma here. And you see the calcium diffuse and flow down all the processes as well. Play it one more time. This is a cell in culture. So this type of behavior you don't see in astrocytes in vivo. As you saw, it's a lot more chaotic and noisy. This is what he said earlier about the wave-like behavior only happening in culture in a cell that's not actually in the brain. So this is not something that we really care about analyzing. What we do care about, though, is using it to verify and validate our flow, because it's clearly, we see what's going on. And so this, thankfully, works out quite well. So we see in the beginning, boom, there's a big flow outward due to that spike that took place in the soma there. So you get a little bit of noise elsewhere, but the dominant flow is represented very accurately when that spike took place. So these are some initial results. So now that we have the data's been denoised, we aligned it, we were able to calculate the flow quite accurately. What I wanted to do is I wanted to try and come up with a way to answer some of the questions that they posed with us earlier. And so one of the things we wanted to know is activity in the processes related to activity in the soma? So if we could somehow find out a way to determine the quantity of calcium flowing up and down along the processes in time associated with the activity in the soma, we might then be able to do some sort of correlation analysis to figure out what's going on. So what I did to define calcium flux is as follows. First, we draw a path along the process. This is something that the practitioner actually draws. This is what they are drawing to determine the flow along this process. What we then calculate is the tangent vector to the flow. This is just some parameterized path by S here. So r prime is the tangent vector. And we get also the gradient vector, or just something perpendicular to the tangent there. And we parameterize that by p. It's just some path q. And then we get the flow also at that location, v of x, y, and t. So the question is, can we somehow use this information to quantify flow that's taking place? Well, those of you who are familiar with fluid mechanics know that flux is defined as the amount of something flowing through a surface per unit time. Quantity, the amount of matter or mass flowing through something. So if we can define the amount of calcium flowing through this plane per unit time, then that would be a good approximation to the flux. So the flux is defined as the inner product between the vector, the flow, and the normal vector to the surface. And that's because, as everybody knows, the inner product is basically a common interpretation of the inner product is how much of one vector is in the direction of another, right? And so here we have how much of this flow vector is in the direction of this vector here. And you just do that all the way along, which is what the integral says, integrating along P, and then that gives you the flux, or the amount of flow going through that surface. Now what I wanted to do, though, is I wanted to go along the whole path, so if you also just integrate over S as well, then you get a nice average representation of the calcium flux going through this path, along this process, in or out from the soma in time. And so ultimately this turns into the amount of calcium moving in the direction of the path. And so this was my technique for trying to provide data or insight into something more quantifiable than just, the ROI, some average in time like that. So that's been working pretty well so far, but they wanted something more. So what they wanted is a way to detect diffusion events. And I realized, using something very similar to that technique, we can do that. So diffusion, also known as hotspots or activity, like I said earlier, they're believed to be locations where the astrocytes connect to neurons. Because I showed you earlier how astrocytes innervate large populations of neurons and modulate their activity. Well, where they connect to it, the neurons actually signal back and forth with the astrocyte, and there's large influxes of calcium in those regions. So if they can find those regions and then do some correlation analysis, you can start getting functional information about it. So the problem, though, is that what you see are a combination of spontaneous spikes that just happen due to some quantum fluctuations, and then some actual significant activity. And they want to find a way to distinguish between the two. So my idea, so here's what a diffusive event looks like. Actually, I'll click play because you got to move the mouse in the silly area. Oh yeah, we're on a long time now. I'll show you real quick. We'll just skip most of it down. So right here, you can see a diffusive event around this spot right here. Bam. I don't know if you guys saw that right there, but that's pretty much what they want to quantify. I'll play it one more time. That little slight pulse right there is what they want to be able to find. So, right, so what I did is I applied optical flow to that, and this is what the flow looks like being quantified over that small little region. So we look at the few frames before and after, and then when that spike comes up, you get a strong diffusive-looking event taking place. There's the frame before, and the frame afterwards, you get this diffusive-looking event, which is what we want to try and quantify. So if we look at the next frame, almost done here, we can show how I use the very similar technique that I used previously to do this. So first, again, as before, we get R of S, and then we get the tangent vector, and then again we have the flow there, except this time instead of trying to determine the flux through it, again, what we do is we actually take the inner product along this entire path and just keep all of those values in a list or a set. And so what this is basically how much of the flow is in the direction of the path at each individual little spatial point along here. And then if we interpolate this, go ahead and click again, we get a curve that looks something like this. And the concept is that if we have positive flux away from the soma and negative flux in towards the soma, those zero crossings along this path should be the place where the diffusion is taking place. It spikes, you get diffusion in opposite directions, it's positive and negative. So if we just use the same technique but actually keep track, instead of integrating, just keep track of all those values, store them in an array, we can simultaneously find the hotspots while doing that as well. So this is actually the latest thing that I've been working on. I actually worked on it when I was in Mexico last week, so it's not completed yet. We have some preliminary results, and it works sometimes and not other times. But ultimately, though, with this information, we are able to do a lot of interesting things, and we're excited about it. So the first thing we can do is spike-triggered averaging, which is basically trying to find out the correlation between when the soma spikes, like a big global event in the cell, and the flow and the individual processes. And then you might be able to draw conclusions like if there's a minimum amount of flow coming into the soma, then the soma spikes. These will give things towards like the astrocyte doctrine, some global rules governing behavior of astrocytes. The detection of diffusive events, that's something they're interested in. Significant activity, like I said, takes place at the neuron junctions. So if we can find out where they happen and do like some sort of a histogram of how many times they've taken place in a certain location, then we can say, those highest ones in the histogram are likely significant and the rest are spontaneous. then we can perform correlation analysis on these certain hotspots. And you might be able to say the activity in this one is somehow influencing this one. There must be some sort of flow or some information taking place, maybe in the same process, maybe in opposite processes, maybe with the soma. And then lastly, this is the exciting part for us, is the hotspots are essentially a point-wise representation of the activity in the processes. So this data is ideal for us for tuning some of the parameters in Evan's astrocyte model. So you guys are familiar with Evan. He's developing an astrocyte model, and it's a point-wise model as well, where the processes are modeled by points and the soma's modeled as a point. And so if we can downsize or quantize this biological data into this point-wise form, we can have something biologically inspired to validate and tune some of the parameters of the model, which is exciting. So that's it.
And I believe we have ample time for a few questions.
Full 3 minutes.
When you're doing the aligning, are you losing any data based on how long you're doing it?
So there's 2 ways to do it. You can either just crop the image based off of the maximum translation, so you don't introduce any new information, you just lose information, or you can extrapolate, which is another technique that's commonly done. I just crop because we're primarily interested in the astrocyte soma itself, so all that periphery stuff we can lose due to some small fluctuations.
So I heard about this empirical mode decompensation technique.
Yeah.
So is that a method that I'm not very familiar with that? Do you think that can be used to do the denoising?
So interestingly enough, it's convenient you brought that up. I was working at the Center for Ocean Atmospheric Prediction Studies with Dr. Wu, the guy who developed it, and I actually used it in my master's research. The problem that I had with it is I'm not able to come up with any sort of an analytic representation of how it's working, and I wasn't able to really quantify in any sort of a better sense of what was going on. That being said, most of the time we just do visual inspection like we did here, but from my experiences, at least on satellite data, the results weren't as efficient as some of the other techniques. Perhaps I just wasn't using it right to explore.
Well, it's not clear how the noisy ensemble decomposition, right? The one we do a lot of averaging in order to
I did the multi-dimensional.
It's not clear how it would distinguish the noise from the noisy part of the signal, where the signal is low amplitude, and the noise contained in the significant part of the signal, which is what he talked about. It is not clear how it would make that distinction because it's based on global functions. So I'm not the author, okay, so I'm not biased. So offhand, my answer would be it would not work. However, it doesn't mean anything. You'd have to ask the author. Maybe something could be done. I have no idea.
Maybe a basic question. So when you talk about noise, you're talking about noise in the image. So is there like biological noise in the sense that the ferret heads was looking at something that was completely non-stimulating? I don't know what that is. Like, is there random stuff? So my question is, would you be overinterpreting this biological noise by ascribing meaning to flashes that have nothing to do with the pattern that the ferret see? I don't know if this is an English question, but
No, it's perfectly legitimate. I mean, ideally, see, right now, we didn't, strictly speaking, to answer that kind of question, right, you'd actually have to do some kind of you basically have to enter into some kind of system 2 signals, right? One is a time signal as which pattern is being showed at what time with the ferret. And then on another signal, you'd have to provide information about the spiking and where they occurred, and then somehow try to figure out whether there's some correlation or not. So at this stage, one would have to say the answer is we don't know. And the fact of the matter is, when you repeat the experiment multiple times, the spiking patterns change. And at some level, they talk about astrocyte fatigue, whatever that means, okay? And in other words, it's like a capacitor. A capacitor discharges, and then you need time to recharge. So while you're recharging, if you feed in the next angle, okay, you get a different result than if the capacitor was charged initially and you feed in an angle at that time, right? So, I mean, the answer to your question is not clear, especially given the fact that you got the blood vessels feeding nutrition, etc., nutrients, and that has its own noise problems, right? So there's a lot of things mixed up in all this.
So what you're describing would be as if the ferret were looking at like a blank screen with no stimulus and there was some activity, and that's some sort of intrinsic biological noise.
Yeah.
So in my noise model, I attempted to sort of model, to quantify for that. I used the Beer-Lambert's law for optical diffusion and added a random like stochastic component to to when that protein I described earlier would emit a fluorophore, or would emit a photon that was able to be seen. So in the noise model, I haphazardly attempted to sort of take into consideration that spontaneous events actually take place.
Now we do have, before the data, okay, before the first angle showed, I believe we got 32 seconds of data which the ferret sees nothing.
Yeah.
So
it's
that
like
could be
background.
construed as biological noise, something, even though we got the data, we never got around to
Oftentimes
actually
people
analyzing.
subtract that from the background because that's like that background noise. that
Also,
you
additional
described.
complication is, since they show each angle for 16 seconds, and then they show each pattern for 16 seconds, and then remove the pattern, right, to let some decays, try to get some kind of reset before you show the next pattern, for repeatability. What they really should be doing is showing 16 seconds, and then maybe for 2 minutes don't show any pattern, to be absolutely guaranteed that there's enough of a pause that all transients have been removed so you can go to the next stage. And while I believe they may have done that, we didn't receive that kind of data. We didn't receive the data. But yes, those are all legit, yeah.
Do you know how long these ferrets were doing these experiments? Because I would just think, that biology seems to be very important, this.
Clearly.
So that essentially the last angle you show that one poor ferret, strapped in that machine,
It's even worse than that. The experiment, we go through all the angles, and then they do that 9 times in a row. That's why it so happens that the spiking, we noticed, and they had noticed already too, that for the first 4 times you go through the sequence of 16 angles, the spikes that occur are relatively strong, right? And the last 4, it's almost like the ferret's gone to sleep, or that all the astrocytes have deactivated, and the spikes have very low amplitude.
Because show the same TV show to a human about 5 times in a row.
There's actually theories about that.
So that's why
There's
we
theories
told them
about that attention
they
involved
do
in that.
for instance they ICA, the independent component analysis, over the entire signal. I told them something wrong here, because if the second part of the signal is intrinsically different from the first part, your regions of interest will be biased by the second part of the signal. So we actually redid the analysis using only the first part where the signal had significant activity, and just threw the second half away. But yeah, those are all good points. And honestly, could not tell you why any of that's happening.
Yeah,
But all valid points.
I was down at the Max Planck Institute over the summer while they were doing the experiments, and so I saw the whole process. It was like a 6-hour surgery ahead of time before they can even get the ferret in there, and then after the surgery, which involves cutting a hole in the brain, injecting a virus, putting in a glass plate for recording. The ferret was only guaranteed to live maybe 7 days after the surgery, and so they go in there and they're in there for like 12 hours a day or more, just recording from the ferret, getting as much data as they can before the poor little creature
Pretty sad.
expired. Yeah, I really wasn't a big fan of that. Whenever they were getting up surgeries, I would turn away and stuff. I don't want to. Don't tell the PETA people, right? But yeah.
Well, we had the great fortune that both Erlebacher and Crock are regularly available around here. So I'm sure that
You buy us lunch, and we'll tell you whatever you want to eat.
So let's thank our speakers again.
Speed through those last few slides, but we got them all.